All right, here we are to talk about energy today in AP Bio, and where we make energy is through the breakdown of sugars inside our body. When we talk about the creation of energy and how it gets used, there's five basic principles that are going to govern the pathways. First, there's a lot of complex chemical reactions taking place. There's not just one reaction for energy from start to finish. There's a bunch of little intermediate things that are happening. Each one of those reactions has a specific enzyme that's going to make it happen. Remember what enzymes do? They decrease activation energy to make sure things occur inside the cell. So each one of these reactions is going to have its own specific enzyme to make sure that the chain reaction keeps going. One of the cool things about this is if I understand how energy is created in a human, I understand how it's created in a dog, I understand how it's created in a tree, a frog, a flower, a snake. All of these different organisms have very similar pathways using the same basic steps. Eukaryotes have all compartmentalized functions because of our specific organelles and us having those membrane-bound organelles that sort of keep everything in its own area. The big organelle that we'll be talking about in eukaryotes is mitochondria throughout the course of this section. And again, those enzymes that catalyze each of those reactions, we can always inhibit or activate them, meaning we can turn them both on and off and sort of control how those reactions are working. So how does this all happen? Well, the first thing we have to understand is that free energy is going to be labeled G on some of these slides that you're going to be seeing. And when we talk about the reactions, there's two basic kinds of reactions that we have inside the body. You have endergonic reaction, and if you look at ender, that should say in, which means energy is going into the reaction. In an exergonic reaction, X means out, so an exergonic reaction is going to release energy that the cell can use for itself. Now, chemical energy inside the cell, you've learned from middle school, is going to be called ATP. And realistically, what the energy actually is, it's not ATP, it's the breakdown of the ATP that releases the energy. So if we start at the bottom here, as ATP hydrolyzes, using water to break it down, you create ADP in a phosphate ion, and you release energy that that cell can use to do any kind of other reaction that's endergonic in itself. This part of the reaction chain is exergonic, releasing the energy. That energy can get used for other endergonic reactions. Now we have ADP in a phosphate ion. That ADP in a phosphate ion takes an input of energy to create ATP. Well, that energy came from exergonic reactions through cell respiration and catabolism. Anytime we break something down, we release energy, and that energy can be grabbed. And this is just this continuous cycle. ATP breaks down in an exergonic reaction that's going to fuel a bunch of other endergonic reactions within the cell. ADP and phosphate require energy to synthesize ATP, and that energy comes from other exergonic reactions going on in the cell. I know it's kind of wordy and kind of chunky, but so understand the breakdown of ATP is always going to be exergonic, and that energy is going to be used for other reactions to take place inside the cell. Putting ADP and phosphate together requires an energy input, and we're going to see in the next couple slides how that actually takes place. Well, when we look at the hydrolysis of ATP, ATP plus water is going to produce ADP, phosphate, and energy. That's a reaction you should know. That produces about negative 7.3 kilocalories. Kilocalories are the body's unit of measuring heat production. If you think of 1 calorie in food is usually a kilocal. There's big C calories and little c calories, and usually we never measure things in little c calories. So big C calories is the same thing as a kilocal. This is negative because there's less free energy in the ADP and phosphate than there is in the reactant, which means there was some energy released. When we look at the molecule of ATP, well, we have our nitrogenous base adenine, we have a ribose sugar, and then we have phosphate, phosphate, phosphate. Look at what's going on with these phosphates. Negative charges here, there's negative charges here, negative charges here. That's a lot of negatives to be putting together. And because we're holding all those negative phosphate groups together, that puts a lot of energy, potential energy, wrapped up in this molecule of ATP. Kind of like if you squeeze a spring, there's a lot of potential energy in that. And as soon as you let it go, the spring bounces back into its normal shape. Well, that's what happens. When this breaks down and we break ATP down into ADP, which is adenosine diphosphate, well, this phosphate breaks off and that releases some free energy that the cell can actually use. So when we look at why there is energy in ATP, it's sort of what I was just explaining, that the phosphoanhydride bond between the phosphate and the oxygen in the ATP molecule is sort of holding all of that energy. And that energy is there because all those phosphates have negative charges, and when we have negative charge on negative charge on negative charge, it's hard to stack those things next to each other. Similar to if you have two magnets and you put the two north poles together, they don't want to stay next to each other. It actually takes some force to get them to stay close. Same thing sort of happening with the phosphates in the ATP molecule. Now, there are other ways to transfer energy aside from using ATP. And, well, ATP, yeah, there is some of this stuff going on, but something called redox reactions. Redox is short for reduction and oxidation. With reduction, whatever the molecule is going to actually gain electrons. And that sounds weird when I say reduction gains electrons, but what you're reducing is the charge. The charge becomes more negative when it gets reduced. So that's why you're gaining electrons, because that's going to make you more negative. Oxidation is the loss of one or more electrons, and that's going to make the molecule a little bit more positive. So you'll probably see a plus sign on something that's been oxidized. And here's a quick saying to help you remember which one is which. Leo the lion says grr. Loss of electron is oxidation. Gain of electron is reduction. So anytime you see that plus sign, that means you've lost an electron, and that's going to be an oxidation. That's kind of your first clue that something was oxidized. Now, here's the wild thing. When something gets oxidized, it's going to reduce something else, and this is sort of the continuum. If I have A and a hydrogen ion, remember hydrogens, we kind of look at those as our electrons and our protons. Well, as A gets oxidized, remember Leo, loss of electron, so this A lost the electron, that H is going to reduce this molecule NAD from up here to NADH. Well, guess what? NADH is going to break down to NAD. As it does that, it's oxidized when it breaks down. It sends that electron to this molecule B over here. And now B was reduced because it gained the electron. Remember, Leo says grr. If you can remember that, hopefully you can figure out which way the redox reactions are moving. And this is just something we have to practice with a little bit. But remember, every time your body breaks something down, you put something back together. You break something down, you put something back together, and so forth. And it just keeps going on and on. And these are what we call coupled reactions, meaning they happen together. The more reduced the molecule is, the more energy that it's holding, because it's got more negative charges in there. So it's just sort of holding that potential energy that when it does get oxidized, it's going to release that stuff. NAD stands for nicotinamide adenine dinucleotide. I will never ask you to write those words out. If you can write NAD, we're good. When I look at the NAD with the plus, that means it's missing an electron, so that NAD plus is something that's been oxidized. When I say NADH, that means it has the electron there, now it's been reduced. And that's sort of how we look at this as we move forward in this section. Well, let's compare the two. The reduction and oxidation of NADH to NAD plus, the oxidation of that, is very, very exergonic, which means it holds a lot of energy that it's going to release. That plays a big role in the creation of energy for us. We use that to fuel some active transport so that we can create a hydrogen gradient in the process of chemiosmosis, which we'll be getting to in a couple slides here. The processes that are going to be carried out in our cell are going to utilize both the ATP and the NADH. Both of these are high energy holding molecules. They have a lot of energy in there in the ATP and the NADH. When I break ATP to ADP and a phosphate, energy is released. When I oxidize NADH to NAD+, energy gets released. And that's going to be really important in the whole cellular respiration that we're going to be getting into in this section. There is another electron carrier that we're going to see in the Krebs cycle that comes out is FAD. FAD plus and FADH, those are other ones that can be used to hold and store energy. They can be used for chemical reactions within our body. Now, how does all this stuff happen? Well, oxidative phosphorylation is something that's going to happen within the cell that's going to create energy. And oxidative means something's going to be getting oxidized, and phosphorylation means something's going to be getting phosphate groups attached to it. Well, that's the coupling of the NADH and ADP turning into ATP. Those two reactions are going to create a lot of energy for the cells, and we're going to see sort of how that works right now. Well, remember this reaction, NADH breaks down to make NAD plus H plus and some electrons and energy. That is the oxidation of NADH. That's an energy releasing reaction. It's exergonic. That's going to fuel an endergonic reaction that we see down here. Energy plus ADP plus phosphate is going to produce ATP, which is a stored chemical energy that your cell is going to use. During chemiosmosis, which is happening in the mitochondria, these two processes are going to be coupled, meaning once this one happens, then the next one, the ATP reaction, can happen. And that's going to create a lot of ATP very efficiently. Well, how does it create ATP? Well, this has to do with gradients and molecules moving. Hydrogen is an ion. Hydrogen is going to have trouble diffusing across a membrane. Why do we think that is? Think about our membrane structure that we talked about in the last section, and hopefully you remember that your membrane is a phospholipid bilayer. Well, in the middle of that phospholipid bilayer are a bunch of lipid tails creating that big lipid lake that surrounds different membranes. Well, do ions diffuse readily across lipids? No. Why not? Lipids are nonpolar, and since this thing has a charge, it's not really going to get through there. So if we can put a bunch of hydrogen ions on one side of a membrane, we can create a pretty big gradient, or our cell can create a pretty big gradient. And if there happens to be a passageway for it to get through, those hydrogen ions are going to want to diffuse from high concentration to low concentration, and we can use that kinetic energy of those ions going through to actually create ATP, which is exactly what our mitochondria does. Now, we have mitochondria, so as a eukaryotic cell, mitochondria in the inner membrane, that's where that's going to happen. Hopefully you remember mitochondria is a double membrane structure. It's got an outer membrane and an inner membrane. The inner membrane has a bunch of folds, which...
Coach Murdoch, please call extension 4204. Coach Murdoch, please call extension 4204.
All those folds are going to increase the surface area and allow more spaces for this proton gradient to diffuse across the membrane. And also in other eukaryotic cells, not humans because we don't have chloroplasts, but plants all have chloroplasts, the green plants. And chloroplasts are also double membrane organelles, and in the thylakoid membrane, they're going to be able to do this chemiosmosis as well. Now, prokaryotes don't have mitochondria because they don't have membrane-bound organelles. They do set up a gradient, though, across our plasma membrane, which is around that outer barrier of the prokaryotic cell, to allow chemiosmosis to happen so that they can make energy similar to the way that eukaryotes make it. It's not quite as efficient as us, and that's why prokaryotes are kind of tiny, because they can't make the sheer amounts of energy that eukaryotes can. Now, look at this word. ATP synthase. What is that? Hopefully you just said that's an enzyme because you see ASE right there. So this is an enzyme that synthesizes ATP. So this is the enzyme or the protein that's embedded in the inner membrane of the mitochondria that's going to cause the hydrogen ions to put together ATP, and we're going to see how that works in a second here. Here is a membrane. We got our phosphate heads, or the blue circles on top and on bottom over here. We got a bunch of lipid tails. And then we got this big old protein right here. I got the F0 unit of the ATP synthase and the F1 unit. In my world, that kind of looks like orange if you sort of peeled it with all the little slices that are inside of it. Well, I have more hydrogen ions outside this membrane than I do inside this membrane. When this ATP synthase is open, which it always is, as long as there's a gradient here, what are these hydrogens going to do? They're going to pew, 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 cruise right through that ATP synthase. And as they go through there, the F1 unit's actually going to spin. And as that thing spins, it takes the ADP molecule and a phosphate that are floating around inside the mitochondria in the matrix, and it smushes them together to make ATP. It's pretty wild how this happens. This happens really fast, about 100 a second ATP molecules. That's a lot of good stuff. That's a lot of energy that our cells can have stored that they can use in other reactions. Well, where do all those hydrogen ions come from? There's something called an electron transport chain, which is a bunch of proteins, and as oxidation of NADH happens, well, when NADH oxidizes, it produces NAD plus and H ions. Those H ions get actively transported into the inner membrane space of the mitochondria, and it's got to use active transport because there's more concentration of hydrogen ions outside in the inner membrane space than there is in the matrix. Where did the energy come from? Well, remember, when you break down NADH, that's an exergonic reaction. So that energy pushes the hydrogen ion outside, and outside the membrane, the hydrogen ions then find the ATP synthase, which they can just cruise right through and sort of get into the cell and create that ATP. Well, why do we breathe then? If anyone asks you why we breathe and they say that oxygen turns to carbon dioxide in your body, that never actually happens. Oxygen plays a huge role though. Oxygen is what we call an electron acceptor. Oxygen comes in at the end of the electron transport chain. As it goes over there, the oxygen grabs the electron that gets passed through the protein chain and it accepts it. The oxygen then becomes reduced, and eventually oxygen matches up with some H ions and creates water inside the mitochondria that your mitochondria will release into the cell. And that's basically why we breathe, to be honest, because without that oxygen there, well, then there'd never be that gradient of hydrogen ions because there'd be nothing to accept the last electron. If nothing accepts that, then you don't have that proton motive force that's created by the gradient of the hydrogen ions inside and outside the inner membrane of the mitochondria, and you never make enough ATP. I'm going to stop right there. Thermogenin, your book talks about thermogenin and how it helps keep infants warm. It sort of turns off the last electron acceptor, but since NADH is still getting reduced, it creates heat. Make sure you check that out inside your Hillis book. We're going to talk about this stuff a little bit more in class and sort of go over how this process actually gets rolling, and hope you enjoyed. I will see you in class.